All right, so I think we're good. So first of all, I'd like to thank the Australasian Brain Stimulation Society for the kind invitation to share some of our team's work today. Many of these studies were collected with our fabulous students, our postdocs, and our collaborators. So before I go any further, I would also like to acknowledge their input into these studies. Okay, let's see. There we go, advancing. So a key theme throughout my entire research career has been how do we maintain active and productive engagement in society throughout life? My PhD was heavily TMS based with the supervision of both Julia Pitcher and Michael Ridding. And during that time, we investigated the age-related changes in corticospinal inhibition and corticospinal excitability and intracortical inhibition. And we did that in older men using a cohort called the Florey-Adelaide Male Aging Study Cohort. And it was really during this time in my PhD that I developed a deep understanding of the non-invasive brain stimulation methods that are really the backbone of this society. So it's really exciting to share some of this work with everyone today. So upon finishing my PhD, I then sought experience working in an exercise physiology laboratory, and we conducted research investigating the optimal exercise patterns for older adults, and we explored the importance of enjoying exercise for long-term behaviour change. Following a 2-year postdoc with that group, I then began marrying my interests in non-invasive brain stimulation methodologies and exercise physiology. So during this time, I was funded, as Bernie mentioned, by both Alzheimer's Australia and the NHMRC, and I was able to establish a team of researchers. So together, my team and I broadly focus on how people use their time and what they eat and how that might increase or decrease their own future dementia risk. So in addition, we undertake acute exercise studies to explore the underlying mechanisms of how exercise enhances neuroplasticity in the human motor cortex. So throughout this talk, I'm going to be discussing a select few studies from our program of research. So I'm going to start with some of the observational studies looking at habitual activity and neuroplasticity. I'm then going to be talking about some of our acute exercise studies and how they provide insight into the mechanisms of neuroplasticity. I'll finish up with a brief overview of our current NHMRC funded project, and then also a bit of a call to action for researchers in the society. So we know that a well-functioning motor system is essential throughout life. It's essential for controlling fine movements such as independent finger flexions, but it's also important for controlling gross movements such as going for a run. So increased age, and we typically see this in people over the age of 60 years, is associated with declines in motor function, including but not limited to a slowing of movement, a difficulty learning new motor skills, an increase in unwanted movements, as well as a reduced ability to correct a movement plan during a movement. We also know that regular engagement in physical activity across the lifespan is really important for both brain and physical health. And certainly there's lots of examples of where regular physical activity is associated with reduced all-cause mortality and reduced morbidity, particularly in late life. We're starting to see some emerging evidence demonstrating the importance of physical activity, particularly in that midlife period, on late life brain health, including the maintenance of both motor and cognitive performance. And we now know that regular physical activity might be able to slow the decline from healthy aging to mild cognitive impairment and then on to dementia. So as I mentioned, our team's main interest is how physical activity might enhance neuroplasticity and indeed protect the brain against some of these late life conditions, including dementia. My interest in the association between activity and neuroplasticity was really sparked in the early 2000s, and this was with some of the first literature exploring the associations between self-report activity measures and response to non-invasive brain stimulation neuroplasticity paradigms. So in particular, one of the first studies in this field was conducted in young university-age participants by Dr. John Cirillo and other colleagues of mine whilst I was working at the University of Adelaide during my PhD. So in this particular study, Cirillo and colleagues recruited highly sedentary individuals. So these people were in the bottom 20% of the population. And they also recruited highly active individuals. So individuals who are in the top 20% of the population. So these people were likely training for marathons or triathlons. And their aim was to explore the differences in these two groups' responses to a paired associative stimulation plasticity paradigm delivered to the primary motor cortex. So the key findings were that at baseline, the input-output curve characteristics when they targeted stimulation for a small intrinsic hand muscle at rest was actually quite similar between the active and the sedentary participants. However, following PASS, they saw a much steeper stimulus response curve in highly active individuals after PASS. And in addition, the active individuals had about a 40% larger MEP response to this PASS paradigm compared to the sedentary individuals. And it's important to note that all these measures were conducted whilst the muscles were at rest. So over the last 15 years, certainly a range of literature has explored the properties of these stimulus response curves, and they've explored them between both young participants and older participants. So my PhD supervisor, Dr. Julia Pitcher, conducted one of the first studies in this area, and she showed that the rate of change, particularly at that lower end of the stimulus response curve, was potentially slower in older participants. However, some of her results also suggested that perhaps the findings, at least in that cohort, might have been driven by the older females. So during my PhD, one of the first studies I undertook was to extend and replicate Pitcher's study. And we studied men only using the FAMIS cohort and demonstrated no age-related changes when men were studied alone. So in both of these early studies, activity levels weren't considered nor explored. So more recently, Stevens-Lapsley and colleagues have then followed up these studies, and this time they've targeted a lower limb muscle during low levels of contraction. And similarly to the earlier studies, they didn't demonstrate age or sex differences in the properties of these stimulus response curves. But they actually did look at some self-report activity measures. So they looked at physical activity in a self-reported way, and they suggested that perhaps some of the properties of the response curves might actually be driven by differences in physical activity. So leveraging these previous studies, some collaborators of mine and I designed a study that aimed to investigate the influence of age, sex, and physical activity on corticospinal excitability. So based on the previous literature in resting muscles, we hypothesized that older adults would exhibit that slower rate of change in the input-output curve, and we thought this would be particularly at the lower end of the curve. In addition, we hypothesized that this change would be greater in older women compared to men. But we did also think that these differences might be ameliorated by sufficient levels of physical activity. So this particular study was conducted whilst I was visiting a laboratory in the US, Dr. Sandra Hunter's lab at Marquette University in Wisconsin. So the experiment was a 2-session cross-sectional study. So the first session involved a familiarization session. So we brought people into the lab and we familiarized them with performing consistent low-level contractions of their lower limb. We also familiarized them with TMS because we were going to target a lower limb muscle. We familiarized them with the double cone coil and stimulations there. And then we sent them away for a week where we undertook a physical activity assessment. And a week later, when they came back into the lab, we constructed those cortical curves, so those input-output curves, using fairly standard TMS techniques. When lost participants were gently contracting their dominant lower limb at 10% of their maximal voluntary contraction. So the target muscle that we were most interested in was the vastus lateralis muscle of the quadriceps. So since all previous studies had used these self-report measures, and we know that self-report measures don't always correlate very well with objectively measured physical activity, in this particular study, we decided to use accelerometry in order to objectively measure activity levels in the participants. So the accelerometer that we used was the Actigraph. This was a hip-worn triaxial accelerometer, and it measured activity during the waking day for 7 days. We then extracted data from the accelerometers as a number of steps per day, and we analysed that through the ActiLife software. So in terms of full data sets, we were able to obtain full data sets from 62 participants. 38 of those participants were old, with a mean age of 70 years. 12 of them were women, and the age range in the older participants were between 61 and 87 years of age. We also recruited 24 young university-aged participants. The mean age was 23 years, and 12 women again, and the age range between 19 and 28 years of age. So similar to the past study, the Cirillo study, we initially wanted to dichotomize our sample into high activity or low activity. So in order to do this, we used the American College of Sports Medicine steps per day guidelines. So if participants were meeting 10,000 steps per day, we considered them sufficiently active, and we grouped them in the active group, whereas those who were not meeting those guidelines were considered low active. And so just a reminder that these data were collected in the US. So actually our sufficiently active participants were very active. So on average, they were achieving about 13,000 steps per day. Conversely, our inactive participants were super inactive. So they were achieving about 4,000, 5,000 steps per day. There were no group differences between young and old in terms of activity levels. However, as expected, the active group were achieving significantly more steps per day than the inactive group. It's also pleasing to note that actually our groups were really well matched in terms of activity levels in this study. So we then compared the properties of these stimulus response curves between age groups, and between males and females, and also across physical activity groups. So neither age nor sex appeared to influence the properties of the stimulus response curves. So for example, both young and old men and women had similar MEP maxes, they had similar slopes, and similar characteristics at that lower end of the curve as well. The key finding here, however, was that the corticospinal stimulus response curve properties were indeed influenced by the activity groupings, but perhaps not in the way that we'd initially hypothesized based on the resting stimulus response curve literature. So I'll just spend a little bit of time orientating you to these figures. So on the x-axis, we've got the stimulator output, and that's expressed as a percent of the active motor threshold. And on the y-axis, we've got the MEP amplitude, and that's expressed as a percent of that Mmax response. So the high active group, so we've got old on the left, sorry, and young on the right. Men and women are combined in these two figures. The highly active group were represented by blue, and the low active group are represented by the symbols that are white. So interestingly, the participants who were meeting those steps per day guidelines actually had a more gradual increase in their MEP amplitude as we increase stimulator output. And a key feature of these data is that all measures were actually collected during contraction and not like the previous literature where they were collected at rest. And so I guess a possible explanation for this is that the participants who are meeting guidelines were perhaps better able to fractionate their muscle activation as we increased stimulator output. So we also compared slope, and we did this at 10%, 25%, and also at the greatest rate of change for the input-output curves. And similar to the previous findings, the participants who were not meeting the guidelines, so in the last slide, they also had steeper slopes at all parts of the stimulus response curve. So these data are shown on the slide separately for old in A and young in B. So together our data here provide evidence that corticospinal excitability in the active vastus lateralis muscle differs with physical activity levels. And it does this more so than with age or sex alone. So insight into these findings provide insight into differences in corticospinal excitability. And we actually then went back to the literature and compared these findings with studies of both strength training and caster mobilization. And so consistent with our findings reported here, in those other literature, we also saw that there was an increase in corticospinal excitability in both upper and lower limbs in response to caster mobilization, so a forced inactivity. In addition, corticospinal excitability appears to be reduced after just even 4 weeks of a strength training intervention. Whilst we need further studies here to fully elucidate whether these differences are due to changes either at the neuromuscular junction, the spinal cord, or neural mechanisms, I'm probably leaning more towards these changes occurring at the level of the cortex. So key take-home messages for this study were that regardless of sex and regardless of age, the participants who are meeting the current physical activity guidelines had a more gradual increase in MEP amplitude. We showed that regular engagement in physical activity can modify the excitability of these corticospinal projections to the active motor limb. And it does this similarly in both young participants and older participants. So following on from that study, when I returned to Australia, I began planning a new piece of work. So as we've already discussed, physical activity may exert its positive effects on the brain through neuroplasticity. And certainly we see in animal models that voluntary wheel running enhances spike timing dependent neuroplasticity, and does this in the dentate gyrus. The Cirillo study provided some limited evidence for us in young adults that habitual activity promotes motor cortical neuroplasticity. However, to date, no studies had investigated these relationships in older adults alone. And in addition, no studies had considered other potentially important daily behaviours. And what I mean by this is no studies had considered the effect of sleep or the effect of prolonged sitting on these associations. So in order to address these knowledge gaps, the overarching aim of the next study that I'd like to talk about was to explore the association between daily activities and neuroplasticity in older adults. So up until this study, all previous work, and certainly the work I've spoken about today, considered physical activity only at the group level. So we dichotomized data into high active or low activity levels. And a key limitation of these approaches is that we're actually unable to consider the importance of other activity and other daily behaviours such as sitting or sleep. So basically, I went into the literature and I had a look at some of the literature that starts to plug these daily time use behaviours into the same model. And we came across some literature that considered that most approaches to physical activity assessment don't actually consider that physical activity is compositional in nature. So what I mean by that is that, and I'll show you this on the right-hand side of the screen using the clock. So every day, whether you're me or you're the President of the United States of America, every day only ever has 24 hours in it. And what we do with that time can either increase or decrease our future health and also our disease risk. So the Australian sleep guidelines suggest that we should be sleeping between 7 and 9 hours a night. Most of us will also spend a period of that time sitting, and so sitting at work or sitting watching TV. And the rest of that day is spent engaging in activities. So those activities might be light in nature, so it might be things like housework or walking to the bus, or they might be moderate to vigorous. So it might be something like structured exercise classes. So we wanted to be able to consider all of these daily behaviours in our modelling. But we actually, in order to do that, we had to take an approach, a statistical analysis approach that's come from geology, so the study of the composition of rocks. And we applied that compositional data analysis approach to the data that I'm going to talk to you about next. Now, I don't have a lot of time in this talk to talk you through how that statistical analysis approach is undertaken, but I have presented a recent review from our team, which you can have a look at if you're interested in understanding the actual analysis approach in more detail. So taking this new compositional data analysis approach, this particular study aimed to determine the association between objectively measured daily activities and motor cortical neuroplasticity in older adults. So we hypothesised that older adults who accumulated higher daily levels of physical activity relative to those other behaviours, sleep and sitting, would have a greater MEP suppression following a long-term depression-like TMS paradigm. So again, this study is observational in nature and cross-sectional. So the participants came into the lab for a familiarisation session, and they also underwent some cognitive assessments. We then sent them away with an accelerometer. This time we used a wrist-worn accelerometer because we wanted to be able to capture those activities across 24 hours. So we captured those activities for 7 days, and we were able to break down the accelerations from the accelerometer into time spent in sleep, which is in grey, time spent in sitting or sedentary behaviour, which is represented in green, and time spent in activities. And we broke those activities down into light physical activity, represented by yellow, and also moderate to vigorous physical activity, which is represented by red. So when the participants came back into the lab, they undertook a neuroplasticity experiment. So we applied TMS to the left motor cortex of 34 older participants, 15 of them were female, and we applied 2 trains of a continuous theta burst stimulation paradigm separated by 10 minutes. We then quantified the response to that paradigm by measuring the peak-to-peak amplitudes of the MEPs after both a single session, a single paradigm, CTBS paradigm, and after the paired paradigm as well. So the first thing we wanted to do, and we'll focus on the graph on the left to start off with. So the first thing we wanted to do was to explore whether our CTBS paradigm did indeed induce the expected LTD-like effects. So I'll orientate you to this graph. On the x-axis, we have time, and we took 2 baseline measures, and then we delivered our first train of CTBS. We took a mid-train measure of MEPs and delivered our second train of CTBS. And then we measured MEPs up until 60 minutes after we'd delivered the stimulation. On the y-axis is a motor evoked potential amplitude expressed in millivolts. So this dashed line represents the average of both baseline measures. So certainly after CTBS, you do see the expected LTD-like suppression of the MEPs at the group level. And this is without considering the effect of daily activities. So then the next step is we wanted to use that CODA approach, that compositional data approach, in order to explore whether daily activities were associated with response to this CTBS paradigm. So what we did is we took time spent in physical activity, and in this case, in order to be able to show this to you on a graph, I've combined light, moderate and vigorous physical activity here. And we took time in sedentary behaviour and sleep, and we then transform these data into percentage of the day. And once you've done that, you can then use standard statistical modelling. So the way that you read this figure is on the x-axis, we've got the percentage of the day that participants spent engaged in physical activities from low to high, left to right. On the y-axis, we've got the percentage of the day spent in sleep from low to high. And on this z-axis, we've got the percentage of the day spent in sedentary behaviour, so those sitting behaviours. And then basically for every participant, you can go and plot their response. So the response in this, the that we're interested in was the MEP amplitude at these post-paradigm measures expressed as a percent of the baseline measures. So a response in the direction that we would expect would be represented by blue on this figure, whereas no response at all to the paradigm would be represented by red. So when we ran these analyses, we did see that daily activities were associated with response to these CTVS paradigms. And interestingly, when you look at the gradient of the response surface that we've applied, you can actually see that gradient shifts from red to blue very neatly along this x-axis. So it appears that more time spent in physical activity at the equal expense of both sleep and sedentary behaviour was associated with a greater response to this LTD-like plasticity paradigm. So if we wrap that study up, this particular study showed us that older adults who achieved days with more physical activity and less sedentary behaviour and sleep had a greater response to an LTD-like plasticity paradigm. Interestingly, when we ran the models with more parts of the composition, we were able to show that the beneficial effects were likely driven by time spent in light physical activity and not necessarily time spent in moderate to vigorous physical activity. And if we take this together, what this tells us is those stealth physical activity interventions, which are starting to gain some traction in the literature, and that these interventions seem to break up sedentary time, they might be a really neat first option for older adults in order to be able to enhance neuroplasticity and maintain neuromuscular function into late life. So moving on from the observational studies, alongside those studies, my team have also been interested in using acute exercise interventions, so single sessions of aerobic exercise, in order to provide insights into the potential mechanisms of how aerobic exercise enhances neuroplasticity. So one of the first studies I conducted in this field explored the effects of acute aerobic exercise on GABA-A mediated short interval intracortical inhibition, and again targeting the primary motor cortex. So the idea for this particular study was developed with Professor Fiona Wood from Western Australia. And Fiona came to visit our laboratory whilst I was finishing up my PhD at the University of Adelaide. Her main focus of research was burns recovery, and she was seeking to understand why her participants seemed to recover so much quicker from injury than many other participants from burns units from around the world. So interestingly, one key difference that they were doing in Western Australia is that they would often get their participants engaged in exercise programs. And as a result, the participants would often leave the burns unit much fitter than when they had entered. So in order to explore the why, we designed an acute aerobic exercise study where we brought young university-age adults into the lab. And these adults underwent cycling at either a low moderate intensity or a moderate to high intensity. And we explored if acute aerobic exercise could modulate SICI inhibition immediately following and 15 minutes after the exercise. So the rationale for investigating the effect of exercise on SICI was that we hypothesized that exercise may be modulating neuroplasticity through altering excitability in the GABAergic system. So for this particular study, we recruited 13 participants, 6 female. We used standard TMS techniques for SICI over the primary motor cortex. That included an interstimulus interval of 2 and 3 milliseconds, as well as conditioning intensities of 80 and 90% of active motor threshold. So excitingly, both the low, moderate, and the moderate high aerobic intensities reduce SICI both immediately after and up to 15 minutes after exercise. And we also didn't see a change in corticospinal excitability. So in order to orientate you to these figures, again, we have time on the x-axis, and on the y-axis, we have that conditioned MEP amplitude expressed as a percent of the test response. So following exercise at both intensities, GABA-A mediated inhibition was reduced for up to 15 minutes. So if we take these findings together, these findings provide us with initial support for aerobic exercise, reducing that GABA-A mediated inhibition. And that may potentially also underpin the acute effects of exercise on increases in neuroplasticity. So since those early studies that we conducted in 2014, there's now a mounting body of literature from a range of different groups that has shown that indeed acute aerobic exercise does appear to modulate neuroplasticity. So my PhD student, Madison Mello, in 2020, conducted a review of this literature, and she showed that in particular, acute aerobic exercise influence non-invasive brain stimulation measures of plasticity, particularly when short bouts of higher intensity interval exercise or longer bouts of lower intensity continuous exercise were employed. In addition, it seemed to be that aerobic exercise was more effective at inducing neuroplasticity when it was used prior to rather than following a brain stimulation paradigm. However, we do need some further literature and some further studies to explore whether these effects occur similarly with different plasticity paradigms, as well as in young, old, and clinical populations. And that was also reviewed recently by this really neat review by Nicolini and colleagues. And so rather than just exploring this effect in older adults and clinical populations, future studies should really also explore whether these improvements or these changes in neuroplasticity also result in improvements in motor learning and or cognitive performance. So before I finish up, each of the studies that I've presented today have really neatly led me to talking about our current research project. So this project is funded for 5 years by the NHMRC, and we're currently coming towards the end of our second year of the study. So in a nutshell, the main aim or the main goal of this research program is to characterise both the best day in terms of daily activities, as well as the best diet for dementia prevention. And then what we're going to do is try and translate that information into a tool that older adults and their primary care teams can then use in order to be able to make behaviour changes and reduce their own future dementia risk. So to do this, we're going to be longitudinally tracking activity, diet and cognition every 18 months for 3 years. Excitingly, as part of the program, both the sites, so we have a site in Adelaide and we also have a site in Newcastle, both of these sites are going to be conducting a secondary mechanism study longitudinally. So in Adelaide, we're planning on focusing on the association between diet, activity and neuroplasticity using NIBS techniques. This time we're going to be targeting the dorsolateral prefrontal cortex. Whereas in Newcastle, they're particularly interested in cerebral artery plasticity or elasticity, and they're going to be exploring this using pulse dot optical imaging techniques. And whilst I can't talk to you about the findings of this study right now, because we're only just, or we're about to reach full recruitment of the participants, I hope that in future ABSS meetings, I will be able to share some of these findings. So finally, I'd like to end my talk with a bit of a society call to action. So as a collective group, certainly since Ritting and Zeman reviewed the literature back in 2010, so we've known for about 10 years that there are a whole range of factors that potentially impact non-invasive brain stimulation measures of plasticity. So these factors range from everything such as gender, aerobic exercise, even time of day. So in my talk, I've also demonstrated that the association between activity composition and neuroplasticity. And recently others like Brenton Hordacre's group have demonstrated associations between obesity and response to neuroplasticity. I'm sure as we collect more data and as other teams collect data, you may also start to see associations between cardiovascular disease risk profile and or diet. I guess my point of sharing this with you today is to just get you to consider how you're characterising and or matching your population or populations. So if you're conducting observational research, how do your populations compare and how do they differ? And if you're conducting clinical trial research, perhaps not only considering how these factors may be influencing your results, but certainly in the space of dementia, multimodal or multi-dimension interventions are really becoming quite important. So in dementia prevention, people will focus on changing activity, changing diet, and changing vascular risk monitoring. And so as a society, perhaps this is a really neat opportunity for us to start looking at how do we change lifestyle, as well as how do we pair that with non-invasive brain stimulation interventions. So finally, we're a small Australian society, so if anyone is thinking of using accelerometry measures in their research, we'd be more than happy to advise you on how to analyse those measures. So before I take your questions, I'd like to acknowledge all the individuals who've contributed to this line of work. So certainly my team at the University of South Australia and my collaborators at both the University of Adelaide, as well as Sandra Hunter's group at Marquette University in Milwaukee, Wisconsin. And finally, I guess none of this work could really be possible without the individuals within this society who review our work, so both in grant review and manuscript review. So I'd like to express that we're really grateful for the positive reviews that you've provided for our work so far. It keeps our team together and it helps us to keep moving this line of work forwards. So happy to take any questions.
Thanks, Ash. That was a really fantastic talk. I just wanted to say very quickly that it was great to start thinking about how geology can start informing better science approaches. And I know that I think a lot of people were really taken by that description. Very cool. So the very first question is really related to that, and that comes through from Kate Hoy, who says, really great talk, Ash. Can you touch on what led you to the compositional data analysis approach you applied?
Yeah, really great talk, Kate. I guess compositional data analysis is really an approach that we need to be taking when our variables are compositional in nature. So we've certainly applied it to time use here, but it can be applied to all sorts of things. So the diet that you've already eaten is a composition because actually everything you've eaten makes up your diet. And you can't actually change that. And so, in fact, when you don't take a compositional approach to these data, you actually end up with a lot of spurious correlations and all sorts of problems with statistics that the biostatisticians tell you can't do. So this compositional approach really solves a lot of those issues. Yeah, we're actually in a meeting yesterday and we're talking about what other data can you apply a compositional approach to. And someone said, oh, well, the brain's a composition, it's made up of all sorts of different structures and all sorts of different parts. So should we and can we be taking a compositional approach to the brain? And it's certainly something that I would suggest maybe we can consider looking at in the future.
Absolutely. So we've got a question now from John Semler saying, hi, Ashleigh, can genetic influence these plasticity effects with physical activity? Perhaps people who are more active have a particular genetic profile that influences plasticity.
Yeah, absolutely, John. This is a really great question and something that I would love to explore. It's not something that I have explored, but certainly, if you think about different polymorphisms in Valmet polymorphisms and that sort of thing. Perhaps it's possible that people with a certain genetic makeup are more inclined to be more physically active. So that would be a really super interesting future research question.
I'm just going to power through because you've got a few questions. So from Nigel Rogash, thanks, Ash. Really interesting talk. Is there any evidence that the effects of exercise or sleep might be nonlinear? For example, could too much high intensity exercise be, oh damn it, Nigel, you used the hard word, deleterious, or is more always better?
Look, I think it's a balance. So too much of anything is not a good thing. So I have colleagues who are not interested in plasticity as outcomes, but they apply these compositional approaches to things such as mental health. And we know that the perfect composition for mental health outcomes looks very different to the best composition for someone whose future job might be an athlete. Someone who wants to play football for Australia, they need to spend more time being engaged in physical activities compared to someone who potentially wants to have better mental health outcomes or better academic performance. So yeah, it's definitely not a more is better approach. It's more about defining the best composition. So we call this the Goldilocks principle. So too much, too little, just right. So we just find that composition that's just right. And there may not be one perfect composition.
So from Brody Hand, thanks for the highly informative talk, Ashleigh. My question is related to the compositional exercise data analysis. I understand the data for this is derived from the accelerometer worn on the wrist. She's got a question mark. And was there a strategy to capture exercise which may not be clear with this method, such as stationary cycling, whereby the wrist and hands are generally static? Thanks.
Yeah, another really good question. So we do have people who work in accelerometry who are super interested in being able to apply accelerations to different behaviours. And we do acknowledge that accelerometry worn on the wrist sometimes doesn't best capture lower limb exercise such as cycling. But we've got these people who are really interested in this. So I'll use my drink bottle if you can see it as a bit of an example. So when you hold a drink of water, you'll hold your drink bottle in a very different way compared to if you're holding a cup of tea. You might hold that very differently compared to if you're holding a beer. And so different people use these accelerations at the wrist in order to be able to attach different behaviours. So there are ways that we can capture different acceleration types using wrist-worn accelerometers.
Thanks, Ash. All right, I think we're going to sneak in one last question. Otherwise, everybody else, feel free to hit Ash up. From Weipang Teo, is there a limit to the number of domains within your compositional analysis?
No, the only limit is being able to show it on a graph. So when you need to be able to show this graphically, you can only limit it to three parts of the composition because you can only plot across 3 axes. You can't go into the 4th, 5th, 6th dimension. But in fact, what we're planning to do with the diet data is we're thinking of breaking the diet data up into a 7-part composition. So we can still run the models on a 7-part composition, but we can't graphically show people those models. We can show them the numbers, but we can't show it on a graph.
Thanks, Ash. Just once again, thank you for such an interesting talk and beautifully articulated for everyone to follow along and understand. So I would just like to remind everybody that right now we have the AGM starting, that the new members will be announced, and that if you'd like to join, well, we'd love you to join, and please turn off your video and mic if you'd like to contribute at any point. So thank you, Ash, and see everyone at the AGM.
Thank you.